Today we'll discuss about the reproduction in CYCAS. In the previous classes we studied about the morphology and the sporophyte and anatomical details of CYCAS in detail. So in this class we'll discuss about the reproduction of CYCAS. In CYCAS the reproduction takes place mainly by vegetative and sexual means. The vegetative reproduction takes place by means of bulbil formation. So what are bulbils? Bulbils are adventitiously produced structures on the plant, and these bulbils are developed or they are formed from the axils of the scale leaf. So in CYCAS you can find foliage leaf as well as scale leaves. We have studied about the scale leaves and foliage leaf. So scale leaves are small structures, they are triangular shaped brown colored structures, and it is from the axils of the scale leaves you can find the bulbils. So bulbils are actually produced from the axils of the scale leaf. That is, they are usually developed in crevices between the leaf bases. So in CYCAS there are persistent leaf bases, and in between the crevices of the persistent leaf bases you can find the bulbils. So bulbils are the adventitious structures that helps the CYCAS for vegetative propagation. So these are the vegetative reproduction structures. So these bulbils help the CYCAS to germinate during favorable conditions. So that is, the bulbils will tide over the unfavorable conditions, and on the return of the favorable conditions it will germinate and produces new plant after falling onto the soil. Okay, always a bulbil from male plant will develop into a male plant, and that from a female plant. That is, the bulbil which is produced from a female plant will develop only into a female plant. Okay, so that is about the vegetative reproduction of CYCAS. Now we'll discuss about the second type, that is sexual reproduction. We know that CYCAS is heterosporous. What do you mean by heterosporous plant? In CYCAS, two kinds of spores are formed. They are megaspores and microspores. Microspores develop into male gametophyte and megaspores develop into female gametophyte. So since the CYCAS is heterosporous, it produces two different kinds of spores. These are the micro and megaspores. So microspores are formed inside the microsporangium. The microsporangium is borne on the microsporophyll, and where can you find the microsporophyll? Microsporophyll can be seen in the male cone. So it is the male reproductive structure or the male cone that produces microsporophyll, and in the microsporophyll microsporangium can be seen, and inside the microsporangium you can find numerous microspores which will develop into male gametophyte. Similarly, there is megaspore. Megaspore is formed inside the megasporangium, and megasporangium is seen in the megasporophylls, and megasporophylls are actually structures which are seen in the female plant. So CYCAS plant is said to be a diploid sporophyte plant. Why is it known as a sporophyte plant? Because it produces spores, and it is a diploid plant. It is a sporophytic plant. Here the predominant phase of the life cycle is a sporophytic stage. That is why we can describe CYCAS as a diploid sporophytic plant. It is also dioecious. Dioecious means there are separate male and female plants. So like I have discussed earlier, the male plant produces microspores and the female plant produces megaspores. So the reproductive parts are arranged in the form of cones or strobilus. The cones are usually seen upon the terminal portion of the main stem. So first we will discuss about the male cone. The male cone is said to be the male reproductive organ. It is produced by the male plant. So what is the structure of a male cone? The male cone is terminal. It is produced at the terminal part of the main axis. It is stalked and it is large and conical in shape. It consists of a central cone axis, and around the cone axis you can find numerous microsporophylls. So the male cone is the male reproductive organ, and it is produced only by a male plant. At the terminal portion of the stem you can find, that is the terminal portion of the stem of the male plant, we can find a male cone. The male cone consists of a central cone axis and numerous microsporophylls which are arranged spirally around the cone axis. And what is the structure of a microsporophyll? Each microsporophyll is woody, wedge shaped, brown in color and leaf like, and these microsporophylls they have a narrow base and expanded upper portion. This upper portion is pointed and sterile, and this portion is known as the apophysis. So, the microsporophyll has a lower or a narrow basal expanded portion and a pointed upper portion. This pointed upper portion of the microsporophyll is termed as the apophysis, and it is this narrow base that helps the microsporophyll to attach with the cone axis. And the microsporophylls are attached to the cone axis by means of a short stalk-like structure. Okay. So each microsporophyle, when we examine carefully, we can find that on the lower surface is termed as the abaxial surface. It is the technical term used to refer the lower surface of a leaf or of a sporophyll. So on the abaxial surface of the microsporophyll, you can find numerous microsporangias, and these microsporangias are arranged in clusters of 3 to 5 groups. So each group consisting of 3 to 5 microsporangia, it forms a cluster known as a sorus. Okay, so microsporangia are arranged in sori around a central papilla, and in between the sori, we can see sori is the plural of sorus, and in between the sori we can find hair-like structure. So it is in this sorus that microsporangia can be seen, and microsporangias are actually sac-like structures. They have a very short stalk and they are surrounded by wall. So the sporangial wall or the microsporangial wall is basically composed of 3 layers. You can find an outer exothecium, a middle endothecium and inner tapetal layer. Okay, and inside the microsporangium numerous microspores can be seen. Okay. So, the development of microsporangium in CYCAS is said to be eusporangiate type. What is eusporangiate type? In eusporangiate type, the microsporangium is developed from a group of sporangial initials. Okay, and the sporangial initials these sporangial initials are actually formed from the hypodermal cells, and it is from the sporangial initials that the microsporangium or the wall layer and the microspores of the microsporangium is formed. The sporangial initials, they divide by a periclinal wall, and they divides into 2 cells, that is the outer primary wall cell and an inner archisporial cell. So each of the sporangial initials divides by a periclinal wall into 2 cells. The outer cell is known as the primary wall cell and the inner cell is the archisporial cell. These primary wall cell, by repeated divisions, it later forms the sporangial wall, and similarly the archisporial cell, by irregular divisions, it later develops into the microspore mother cell. So each microspore mother cell is said to be diploid, and then it undergoes meiotic division and forms 4 haploid microspores by meiotic division or reduction division. Okay. And what about the microspores? The microspores or pollen grains are globular, they are uninucleate, unicellular and haploid structure. And when you study each microspore in detail, you can find an outer exine wall and an inner entine. Okay, so each microspore has 2 wall layers, an outer exine and an inner entine. The entine encloses cytoplasm with a nucleus. At the center, you can also find a large nucleus. So this is about the male reproductive structure. So the male reproductive structure or the male reproductive organ is known as the male cone, and the male cones are produced exclusively by the male plant, Each male cone is a stalked, large, conical shaped structure. It is produced in the terminal part. It has a central cone axis and around which microsporophylls are arranged. And the tip of the microsporophyll is narrow, short, and it is so termed as apophysis. The basal portion is wider, and the microsporophyll bears microsporangia on the lower surface, and each microsporangia or the 3 to 5 microsporangial groups they are arranged in a cluster known as sorus, and inside the microsporangia you can find numerous microspores. Each microspore has two wall layers, outer exine and inner endine. The microspores are also termed as pollen grains. Okay. So, the pollen grains are nothing but microspores, and these microspores are globular, uninucleate, unicellular and haploid structure. Okay. Now, let us discuss about the female reproductive structure or female cone. Okay. Actually, in CYCAS there is no true female cone or female strobilus. Here, the female reproductive organs are more commonly termed as megasporophylls, and they are not commonly termed as female cones, because here the megasporophylls are not organized into cones. Okay, and the megasporophylls are arranged spirally around the stem apex, just like a foliage leaves. So, in the case of CYCAS, we know in male cone also the microsporophylls are arranged spirally around the the central axis, just like that. And here also the megasporophylls are arranged spirally in acropetal succession around the stem apex. Okay, just like a normal foliage leaves, and they are very large. They are produced in large numbers than the foliage leaves, and they appear like a rosette or a crown like fashion. So each megasporophyll is leaf-like, and the megasporophylls are densely covered with brown hairs. Okay, the megasporophylls are considered to be modified leaves. They are flat, dorsiventrally flattened structure, and they measure only about 15 to 30 centimeter in length. Okay, so the approximate length is about 15 to 30 centimeters, and each microsporophyll, it contains a lower portion which is termed as the stalk or petiole, that is very narrow, just like a normal stalk. Then there is a middle expanded portion which is the ovule bearing portion, and then there is a tip which is now termed as the upper leafy lamina. The upper part is usually sterile, and the shape of the upper part varies from species to species. In the case of CYCAS revoluta, the upper part is dissected. Okay. And in the case of CYCAS rumphi, the upper part bears only short spines. So likewise, from one species to other, the different species of CYCAS shows variation in the upper part of the megasporophyll. Okay, so the middle portion of the megasporophyll is the fertile portion, and it bears ovules. So ovules are produced in two rows on either side of the megasporophyll. So at the central portion of the middle portion of the megasporophyll, on either side we can find ovules, and ovules are arranged in two rows, one on each side. So ovules of the two rows may be oppositely or alternatively arranged, and each ovule is a yellowish or orange shape structure, orange colored structure, and they are shortly stalked, oval and smooth. All the ovules will not develop fully. Some of the ovules, they may remain unpollinated, and finally some of them may abort. So the ovules of CYCAS is said to be orthotropic, that is straight ovules, and it is unitechnic also. The stalk is very short. Now we'll explain about the ovules of CYCAS. The CYCAS ovules are the largest in the plant kingdom, and these ovules are sessile, that is they lack a stalk, and if stalks are present, then the stalks will be very short, and the ovules are borne on horizontally on the two sides of the megasporophylls. The ovules are green in young condition, and they are protected by hairs also, but at maturity their color slowly changes from green to orange or reddish in color. And the ovules are formed on the two sides of the megasporophyll. Megasporophylls are nothing but the modified leaf-like structure, and the ovules are said to be orthotropous ovules. So orthotropous means They are straight ovules and they are unitechnic also. Unitechnic means the ovule is protected by only a single integument. If there is only single integument, then the ovule is known as a unitechnic ovule. Okay. So this single integument, it is composed of three layers. The outermost layer is termed as an outer fleshy sarcotesta, which is usually green or orange in color. Then there is a middle yellow stony layer, which is also termed as sclerotesta, and then there is an inner fleshy layer. So the outer layers and the innermost layers are fleshy, whereas the middle layer is said to be stony layer and is termed as the sclerotesta. The outer and fleshy layers, the outer and the innermost fleshy layers are provided with vascular strands or vascular supply, whereas the middle stony layers lacks any vascular supply. So the ovule thus consists of integument, then there is a short stalk, and the upper portion of the ovule you can find a very small opening which is termed as the micropyle. So micropyle is seen at the uppermost portion, and the nucellus, you can also find a nucellus. The nucellus lies just below the integument. So just beneath the integument you can find a nucellus. The nucellus encloses the megaspore or embryo sac. Then the megaspore nucleus undergoes repeated division and it forms the endosperm or female gametophyte in the later stages. In some cells in the nucellus towards the uppermost portion, certain cells dissolves and forms a cavity which is termed as pollen chamber. So the pollen grains are usually received in the pollen chamber after pollination. Just below the pollen chamber you can find an archegonial chamber, and just below the archegonial chamber 3 to 6 archegonia are present towards the micropylar region. This archegonial chamber is also filled with a fluid. Then in the central region of the nucellus, one of the cell, it differentiates into a megaspore mother cell. So what is nucellus? Nucellus is the fleshy mass of diploid tissue that is contained in the ovule, and the nucellus is actually encased by the integument. So the nucellus at the upper portion you can find a very small opening which is termed as the micropyle, and this micropyle is the region that allows the entry of the pollen grains. So in the central region of the nucellus, one of the cell differentiates into megaspore mother cell. Since the nucellus is composed of diploid cells, the megaspore mother cell is also diploid, and this cell, it can be identified by, that is the megaspore mother cell can be identified by dense cytoplasm, larger size and prominent nucleus. This megaspore mother cell will undergo reduction division or meiotic division and forms 4 haploid megaspores. So the 4 haploid megaspores are formed as a result of reduction division from a single megaspore mother cell which is diploid. The lowermost megaspore only acts as the functional megaspore, whereas the upper 3 megaspores, it degenerates and provides nourishment to the functional megaspore. So the ovule of CYCAS is said to be the largest in the plant kingdom, and this ovule it can be visible with our naked eye, and the ovules are said to be orthotropous ovules, that is straight ovules. They are unitechnic and they are the largest in the plant kingdom, and some species of CYCAS produce ovules which are 6 to 7 centimeters in diameter. Then young ovules are usually green in color, protected by hairs, and at maturity the hairs will disappear and the ovule will become orange or reddish in color. So the ovule encloses nucellus, or ovule consists of nucellus, and just within the nucellus some cells functions as megaspore mother cell, and then it differentiates into large. It can be differentiated by the large size, dense cytoplasm and prominent nucleus, and these megaspore mother cell undergoes reduction division and form a linear tetrad of 4 megaspores. So from the central region, a single megaspore mother cell undergoes meiotic division to form 4 megaspores, and the megaspores are haploid. The lowermost megaspore is functional, whereas the upper 3 degenerate to provide nourishment to the functional megaspore. So this is the structure of the ovule. It's a very simple structure. Now the next is about the gametophyte stage.